Hi, I'm Jake. I'm presenting my talk entitled Widespread Redundancy in Omics Profiles of Cancer Mutation States. So here's kind of the overview of what we're going to be talking about. The general theme is sort of functional omics in cancer. I'm going to funnel that down to sort of a specific prediction problem that I think is a good proxy for sort of functional omics. And then I'll talk about our actual current work. We're doing kind of an omics bake-off where we're comparing different omics types in TCGA for this problem. We're going to see how well our conclusions generalize to survival prediction. And we're going to try to build multi-omics models and see if we can outperform the individual omics types. So that's the general theme. So cancer is a disease of genetic alterations. Normal cells sort of proceed towards becoming precancerous based on a single sort of driver mutation. And then mutations sort of start to accumulate in the cells. Some of those might be drivers that sort of drive the cells further towards developing into a tumor or a malignancy. And some of those are just sort of passenger mutations that don't have any real effect. And particularly in the case of the drivers that are sort of actually driving cancer development, we can think of them in the context of cell function. So we can think of sort of the information flow going from DNA mutations to the cancer cell phenotype. But we all know that it's a lot more complicated than this. We can think of information as propagating from the mutations sort of through gene expression, through protein abundance, and eventually to the cancer cell phenotype. But it's even more complicated than this when we think about things like epigenetics. Sort of signal might propagate from the mutations through DNA methylation or histone modifications and things like that, which can then subsequently affect gene expression and the rest of it. And we can even think of DNA mutations in genes like DNA damage repair genes that can change patterns of DNA mutation themselves. So there's a lot of different ways that information can sort of flow from a mutation causing cancer to change the cell phenotype. But the salient point here is that cancer mutations, if we look at mutations that have sort of a big enough effect size or that are really driving cancer, they should leave a detectable functional footprint that we can find, ideally in most of these sort of data types. So one way to get at that is to sort of flip the problem on its head. So we can have, in sort of a prediction setup, we can have the labels be the presence or absence of a particular genetic alteration. And we can have the features in our predictor be the sort of downstream functional readouts. And in our case, we're going to consider expression, DNA methylation, protein abundance, microRNA, and mutational signatures as the sort of downstream readouts that we're using as predictive features. We can take this from TCGA. We can use processed level 3 pan-cancer data from TCGA to define our labels, our mutated or not mutated labels for each sample. We can use point mutations plus CNV info, also from TCGA to do that. And we can use this to ask a couple of questions about a particular gene. In this case, we can say, for a particular mutation or for a particular gene, can we detect a functional signature using any of these data types? Or we can also ask questions about the data types themselves. We can say, for this data type, can we detect a functional signature, for various genes, right? So we can use that to compare the information content of different data types. But why might we want to do this? Well, one reason is just as a sort of benchmark for cancer-related information content. So we can ask the question, what's the biggest contributor to predictive signal in this case? Is it the data source? Is it the mutated gene? Is it both? Is it neither? We'll see. And downstream, this can sort of lead us to ask questions about study design. So we can ask sort of, when are different data sources redundant? When might they provide unique information? If you're doing a cancer study, which of these sort of different data sources might you want to select? So that's one question that we can ask using this sort of setup. So like I said, we're going to do kind of a bake-off first. We're going to ask which data type is best for this problem out of the ones available in TCGA. So we started off by just comparing gene expression and DNA methylation in particular. The reason for this is that we take samples that are sort of profiled for all of those data types. So in this case, this gives us about 8,000 total samples when we limit it to only expression and methylation. We're using 27K and 450K methylation array data from TCGA, and we're also using RNA-seq data, of course. And as sort of target genes to define our labels, we're using about 280, cancer-related genes from these 3 sort of review papers. They're all sort of, a lot of them are strong drivers or genes that are sort of shown in the pan-cancer context to be very sort of cancer relevant. To evaluate performance, we're splitting the data up into train and test folds. We're going to do that using stratified cross-validation. So our train and test sets have equal proportions of each cancer type in TCGA. And we'll do that 8 times, so 4 folds and 2 different random seeds. As a metric, we're going to look at AUPR, area under the precision recall curve. The reasons are that it handles continuous output well, which is given by our elastic net classifiers. And it also works well for imbalanced labels. So a lot of these genes, are only mutated in a small subset of samples. And looking at AUPR tends to give more meaningful comparisons in that case. And as a baseline, we're going to look at a model with permuted mutation labels. The reason is this sort of controls for cancer type. It controls for the predictor that's only using cancer type information to sort of predict whether a sample is mutated or not. So we want to look at sort of signal above that baseline. And we can look at these sort of volcano-like plots to sort of quantify this. So in these plots, each point is the results for one gene and one data type. We have gene expression and the two methylation data types here. On the x-axis, we have the mean difference between the true model and the baseline that we just talked about with the permuted labels. And on the y-axis, we have a p-value for a t-test that's sort of comparing the 8 different cross-validation folds between the true model and the shuffled model. So for well-performing genes or well-performing classifiers, we want to be up and to the right. And we can just count the number of genes that are sort of above a cutoff. So for gene expression here, we can see that we have 113 genes above that cutoff. For 27K and 450K methylation, quite a few less, so 62 and 77 respectively. So we can see here that we sort of generally see better performance for gene expression across the whole gene set. We can also summarize using a box plot where each point is a gene, each column is a data type. And again, we're looking at difference above the shuffled or the permuted baseline on the y-axis. And we can again see the gene expression. The mean performance for gene expression is a little bit higher. So what if we want to compare more omics types? We can use, in addition, we can use protein abundance data in the form of RPPA, microRNA, and mutational signatures, in this case, single base signatures. And so again, we're using samples where all of those data types are measured. That gives us about 5,000 total samples. So it's quite a bit fewer samples in this case. We can do the same thing. We can do the same kind of volcano plots, and we can just count the points that are sort of up and to the right. And for gene expression, we see about 80 genes, 27K and 450K methylation, not quite as well. RPPA is sort of comparable to the methylation data types. MicroRNA slightly less, and mutational signatures is not particularly effective for this particular prediction problem. And we can also look at a box plot that summarizes across all genes. So again, y-axis is difference against the permuted baseline, and each column is a data type. And you can see that mean performance for gene expression is a little bit better. So we also wanted to ask if we use these different omics types to predict survival, do we see sort of similar redundancy, similar results? So we did, in this case, we did PCA preprocessing for all of the omics types to sort of get comparable signal per feature for each of them. We used elastic net Cox regression with covariates for age, cancer type, and mutation burden. And we used a baseline predictor that only uses non-omics covariates, which tends to be pretty hard to beat in previous studies. And as a metric, we're using concordance index. Higher is better in that case. And so in this case, the x-axis is the number of principal components and the y-axis is the concordance index. This gray line is the covariate-only baseline. So there are kind of two takeaways here. One is that there's not a huge difference between the different data types. And two is that they're all pretty close to the covariate-only baseline. So it's hard to beat just using clinical covariates in this case. So again, we can throw in all data types. In this case, the covariate-only baseline does a little bit worse on the sort of reduced set of samples. But we can again see that all the data types are kind of clustered pretty closely together. So there's not really a right choice here. They all kind of give comparable performance in this case, sort of similar to what we saw for mutation prediction. Okay, so now we want to return to the mutation prediction problem. And we want to ask if we can combine data types to improve performance. So instead of using, in our previous experiments, we just used individual data types on their own. We want to ask if we can combine data types to do better than those single omics models. So in this case, we're just looking at the most predictive data types, which were expression and the two methylation data types. For each of them, we took principal components to make sure that the information content per data type is sort of equivalent. And to form a multi-omics model, we're just going to concatenate the different data sets. So we're going to just take expression and methylation, form one big feature matrix out of all of them, and train an elastic net model on those. And we looked at all pairs of those data types plus the combination of all three. To give us a big sort of feature matrix. And in this case, we focused only on 6 driver genes that did pretty well in our first set of experiments. So those are listed here. If you study cancer, you probably recognize a lot of these. These are sort of genes that are known to drive cancer in sort of a pan-cancer context. And so here, the green boxes are the best single omics model type for that gene between expression and methylation, and the orange boxes are the best multi-omics model for that gene. So the pairs of expression and methylation, and then all three combined into a single model. And if we do paired pairwise t-tests between the single omics and multi-omics performance across the 8 cross-validation folds, we see that none of these differences are actually significant at a cutoff of 0.05. So in none of these cases is it more effective to train a multi-omics model than the best single omics model for that gene. So just to summarize what we've kind of learned here, we see in general that gene, not data type, is the primary source of variability. So gene expression in general tends to capture the most signal. It's probably the best default for any given gene. But many genes that are predictable using one of the ohm are also predictable using multiple ohms. Or in other words, mutations with strong functional signatures tend to perturb all of the ohms to some degree. Just to sort of emphasize this, here's another visualization of the same thing. So in this case, columns are genes, rows are training data types. A gray dot on that gene in that data type means that model was significantly better than the baseline, and a black dot means the same thing, and it's equivalent to the best data type. So I guess the takeaway here is that there's a lot of sort of vertical bars, which is sort of saying that you can train a good classifier for that gene using a bunch of different data types. So if you look at, say, BRAF or TP53, you can see that for those sort of strong pan-cancer drivers, you can train a gene using just about any data type. You can train a model, using just about any data type, and you can get pretty good performance. So these sort of strong cancer drivers are leaving signatures throughout all of the different omics types. And that was kind of an interesting result that we noticed. We saw that survival prediction was similar between the ohms. There's not much difference between omics types. And in general, it tends to be fairly difficult to beat a baseline that is just using age, mutation burden, and cancer type, right? So it's generally fairly difficult to beat that covariate only baseline. And we saw that for this problem, for these 6 genes, multi-omics integration doesn't seem to help that much. There was no statistically discernible difference between the best single omics and multi-omics model for these genes. We also saw the most useful omics type varies with the target gene. I didn't show those results, but those are in the paper. And we also used a relatively simple method here of concatenating data types. We tried elastic net. We also tried a 3-layer neural network in this case, and we did not see any improvement there. So maybe there's room for improvement using more complex methods, but we didn't see it in this particular case. We also sort of present this as a resource for experimental design. So say you're studying a particular gene and you want to know sort of in a cell line or whatever, which data type readout is the best. You could look at this table in our manuscript and you could say, for BRAF, we see pretty good performance using gene expression and 450K methylation. So maybe we should pick one of those. So this can sort of be a resource for study design in the future. I want to acknowledge people in my lab, in particular my co-author, Casey Green, my advisor, also our collaborators, Brock and Maria, who helped a lot with sort of developing the methods here and making sure that the analysis works well. And also my thesis committee. We published a paper recently in Genome Biology. There's a link here. Code and analyses are available on GitHub. And I'm happy to talk about this stuff if people have questions. Thank you.